All right, we might have a few more folks joining us, but we'll go ahead and get started. Good afternoon and welcome to the Spring CESU, that's Cooperative Ecosystem Studies Unit, webinar series. My name is Lisa Gerloff, and I am the Executive Director for the Rocky Mountains CESU. This year, with our series, we are joined by the Colorado Plateau and the Desert Southwest. Each of our CESUs selected a project and a researcher to showcase the diverse range of good science that is accomplished through our regional networks and the impact it has on management decisions. The Rocky Mountains kicked off the series March 12th with a presentation by Haley Johnson, a University of Montana graduate, recent graduate, drawing on research from Yellowstone and Stephen Mather Wilderness. Haley explored how social science can manage growing recreation demands on our public lands. The Colorado Plateau was represented March 25th by Northern Arizona University's Tumulin Sankey. In her presentation, Tumulin demonstrated how LIDAR-derived fuels monitoring data are essential to evaluate wildfire risk, plan management activities, and evaluate fuel treatment effects. In a few minutes, I'll share a link to our seminar webinar page in the chat. From there, you'll be able to find links to those two presentations, and tomorrow we'll add this presentation to that page as well. Today is the Desert Southwest CESU's Day, and I am happy to have Mallory Davies with us. Mallory is a researcher working on her PhD at Colorado State University and is a lab manager for Dr. Catherine Stoner's lab at the University of Arizona. Mallory will be discussing how migration, diet, and disease monitoring are informing conservation strategies for bats and desert ecosystems. Mallory will speak for about 35-40 minutes, and we'll have plenty of time for questions afterwards. I'll remind you, but please put your questions in the chat box. With that, I welcome Mallory Davies, and I turn it over to her.
Hi, everyone. Thank you for joining me today. I'm going to synthesize a decade of work on long-nosed bats in the desert Southwest and show how diet, migration, and disease risks are connected. But first, I'd like to acknowledge Dr. Catherine Stoner and her lab, who have been conducting nectar bat research in New Mexico since 2014. It is a privilege to build upon the foundational work initiated by Scarlett Sellers, Rachel Burke, and Teresa Laverty, and to continue our collaboration on this impactful project. And I would like to acknowledge the current lab members, Madeline Von Rutz and Matt Flores, whose efforts continue to contribute to the conservation of these important species. Our research focuses on the three nectivorous long-distance migratory bats that persist in the U.S. More specifically, with support provided by the Bureau of Land Management, we are monitoring the New Mexico population as part of the U.S. Fish and Wildlife Service post-delisting monitoring plan for the lesser long-nosed bat, Leptonicterus yearbook 1A, and we are monitoring for the presence of the federally endangered Mexican long-nosed bat, Leptonicterus nivalis. Our work takes place in the Sky Islands of Arizona and New Mexico, a major biodiversity hotspot supporting over 30 bat species, including migratory nectar bats and many species vulnerable to white nose syndrome. The recent arrival of PD in this region changes the ecological context considerably. These bats move through highly connected roosting and foraging networks, often overlapping with white nose susceptible species, raising important questions about how migratory connectivity may intersect with disease spread. That intersection between movement ecology and pathogen invasion risk is what motivates this work, and it reflects how a decade of ecological research has led naturally into our current surveillance efforts. Our work in this region began in 2014 when Catherine Stoner joined New Mexico State University and launched a research program focused on migratory nectar bats in the desert Southwest. Early graduate work by Rachel Burke has helped define the resource landscape and support the nectar corridor hypothesis, while Scarlett Sellers expanded that foundation by examining population-level diet dynamics. That work grew even further through Teresa Laverty's postdoctoral research while at Colorado State University, which advanced our understanding of migratory connectivity and helped establish a collaborative pit-tag network across the region. Since 2020, my work has built on that foundation by integrating phenology, diet, movement, and population monitoring to investigate the drivers of migration and, more recently, the implications for emerging disease risk. Alongside collaborations with Madeline Van Rotz and Ed Flores here at UA, we are working together. These efforts moved from studying pieces of the system to studying how the system functions as a whole. So through these efforts, we built a framework for examining how ecological processes operate across scales, from local resource phenology to regional movement patterns. Linking these layers allows us to ask how temporal and spatial variation in resources shapes both individual behavior and population level dynamics. So first we'll start with movement and resource tracking. One major accomplishment that we recently has been showing that long-nosed bat migration timing is closely linked to nectar availability rather than fixed seasonal timing alone. So long-nosed bats are migratory bats characterized by resident and migratory subpopulations. Some populations migrate over 1,800 kilometers north each spring from south-central Mexico to the southwestern United States, following a hypothesized nectar corridor of keeping their cacti and agave species. So here's a really nice map that came out in a recent publication showing these two populations. Subpopulations are not genetically distinct, though. There is movement between the two. But the migratory population in the winter, they have their mating roosts down here in central Mexico, and there's some down here in southern Mexico as well. In the springtime, pregnant mothers move north to maternity roosts, some in Sonora, over in Arizona, where they give birth to their young. When they're young or volant and can fly, they move over to transition roosts in southeastern Arizona and southwestern New Mexico and northern Mexico, and in Texas, before they migrate south again in the fall for the winter. So Rachel Burke's master's work tested the spatial temporal nature of the nectar corridor hypothesis using independent data and determined that species richness of food plants is a major driver for migration and distribution for all three nectarverse bats in the United States. And more specifically for Leptocnectaris yerba, Burke's findings indicated a shift in relative importance of specific food plants throughout the year, supporting the theory that nectarverse bats undergo seasonal shifts in habitat selection based on available food sources, also known as resource tracking. So this figure is a seasonal distribution model for lesser long-nosed bats. The map shows habitat suitability based off of nectar food availability. The green shows low suitability, yellow, and then it goes yellow to red with high suitability. So here in the winter, suitability is more concentrated. In the spring, it's a bit more spread out. Summer becomes concentrated again in certain regions, and then the fall becomes spread out throughout the range. So as you can see, so just pointing out, in the spring and fall, the red is a bit more spread out, it's not as specific. It indicates that distributions of these bats were most uncertain during their migration periods, which suggests that factors other than the species richness of food plants may influence migration patterns during that time. So to further investigate this relationship, we focus on the long-nosed bats population in the northern region of the Chihuahuan Desert ecosystem and the boot heel of New Mexico. This is one of the areas where the bats distribution is most uncertain. The arrival of the bats to this region, indicated by the blue star, is thought to coincide with the regional flowering of agave species, the only known plant nectar food source used by long-nosed bats in New Mexico. There are two dominant agaves in this region. It's agave palmeri which is the most abundant agave species that persists at lower elevations. And at higher elevations, there's agave parrot that can be found growing in sparse patches near mountain peaks. Regional droughts have disrupted the bat agave synchrony, contributing to declines in agave populations and potential mismatch between bats' presence and the agave bloom. So this happens because agave blooms timing, it begins bloom peak in July through mid-August in New Mexico, and the bats tend to arrive in June, but then depart late October. So not really, they are here at periods when the agave isn't blooming. Regional droughts have disrupted this timing and has led to declines in agave population, which is potential for why this mismatch is occurring. And resources are becoming less available. So there's reduced nectar in late spring and early fall, which indicates since the bats are still here, they may be using other resources. So to evaluate the risk of phenological mismatch and potential impact on longnose bats populations, we ask two main questions. How does agave nectar availability influence longnose bats' arrival and departure in New Mexico? And how does the diet of lesser longnose bats vary seasonally and annually in relation to nectar availability and regional drought conditions? We'll start with the first question. How does agave nectar availability influence longnose bats' arrival and departure in New Mexico? We came up with three hypotheses. So our null hypothesis is the fixed migration hypothesis, which states that longnose bats' movement patterns are innate migratory behaviors associated with ordinal day, resulting in low variation in arrival, departure dates, and seasonal abundance. Alternatively, bat movement patterns are driven by agave nectar availability in New Mexico. We would predict longnose bats will track the phenology of agave to exploit this ephemeral resource. So we would expect a shift in timing of occupation relative to agave phenology. And third, the lesser longnose bat movements is driven by climate patterns in New Mexico. So we would predict that climatic cues such as relative humidity and temperature thresholds will determine longnose bats' movements. To test our hypotheses, we monitored longnose bat populations and used our agave nectar availability data that we collected. That will go over in the future, but we collected that within a 50-kilometer radius of the roost. The Big Hatchet Mound Roost is located in a wilderness study area in southwestern New Mexico on unceded Chiricahua Apache land. Big Hatchet Mound Roost is the largest known roost site in the state of New Mexico. It's believed to be a late summer transition roost where longnose bats, mostly female in their young state prior to their migration south. In 2019, we installed an Access Communications Q1942E thermal camera and recorded nightly video counts. We use these thermal videos to monitor the longnose bats' weekly population size and departure and arrival times. So to be able to tell if a longnose bat was in it, it's a multi-species roost. We're 10 different species there. So we look for T-shaped forearm musculature. So you can see here is a longnose bat. And there's kind of this T-shape. Their arms are so strong from being able to do the long migratory routes that they have this prominent forearm musculature that we can actually get a heat sense off of. So that you'll see their arms sticking out. There's their legs down there, and that's their head. We also look for a leaf nose. So they have a nose that kind of sticks out a bit. And can be identified where we don't have any other philostomid or leaf-nosed bats in this case. And then there's no tail membrane, so you can see there's a gapping between the legs there, and that helps us differentiate them from the nectar bats. However, this is really hard, and there's a lot of observer bias, so our data on this is still inconclusive, and we're currently building a Python model that'll automatically count the bats for us to help even out observer bias across every video. And here you can see this is how fast the bats would fly through the video, and we'll have to try to count them as they fly through that fast. So we go frame by frame, count each individual bat, try to make sure that we get them as they're coming back in, or not double count the same individual. So we're like, okay, that bat flew out, but it flew back in. So this is tracking the same 2 individuals flying in and out of frame multiple times. Located inside the cave and outside the cave since 2020, we have temporarily deployed 2 temperature humidity monitors and we've also deployed an additional one inside the lower room that the leptos occupy. Also at the base of Big Hatchet, we installed a weather station that records temperature, relative humidity, precipitation, wind speed, and wind direction. Long-nosed bats, as I stated earlier, are known to forage up to 100 kilometers in one night. Therefore, to monitor nectar food availability, we surveyed agave phenology and density within a 50-kilometer radius of the Big Hatchet roost, which is represented by the white circle here around the blue star on the map, which represents Big Hatchet. We established and monitored 5 phenology transects from 2020 to 2023. Transects were visited weekly to track individual plant phenophase. In total, we monitored 730 individual plants, 648 were agave palmeri, and 82 were agave perii. Looking at just the agave palmeri that were monitored, we found that agave palmeri nectar availability peaked in mid-July. Agave peaks were approximately 50% of all the plants that flowered that season were in bloom for about 6 weeks each year. And there's a variability of this peak between years of plus or minus 14 days. So that's a pretty significant difference when you think about plasticity of movement. Looking a bit closer at this figure, this is the total number of individuals that were blooming throughout the season and the percentage of those individuals that were open in a given week. So here in the beginning, there were 0% before June 6th. Then we had our first few plants peak. At the peak, we considered peak over 50%, so it would be above this line, where at least 50% of the individuals that bloomed that season were currently in bloom. And then they all kind of tail off around the beginning of end of August, beginning of September is when most of the plants are done blooming. And the last few are blooming towards mid-September each year. So we would anticipate long-nosed bats abundance to be relatively low in this period in late May or early June, when the agave are just beginning to bloom. We'd expect to have maximum or larger populations when the agave nectar availability is at its peak. We expect the long-nosed bats populations to decrease like September, late August, early September, when the bats or when the agaves are almost done blooming, or for the most part done. And we wouldn't really expect to see any bats mid-September on. Now we're still having issues with the thermal camera counts, which is why we're developing the model, but we did collect fecal samples weekly from a tarp that was placed near the entrance, and we were able to get a relative population count off of that number using total number of fecal samples collected in the season and looking at the percentage of samples collected in that given week to see what the peak was. And we used the week with the highest number of samples collected, actually, and then we went from there for week by week to get a relative to the peak population. So it would scale with the agave phenology. And as we can see, in 2020, there was a peak at the early season, but we believe that is due to agave perii will actually have a peak bloom in early June. So we believe that was a good agave perii year, and that's why we saw a peak in fecal counts then. But overall, looking at the relative peak for each season or each year, we do see that the peak overlaps the peak bloom, or the population peak overlaps the peak bloom of agave. So when we overlay the roost population size relative to the number of pupils collected annually, we found a similar shift in roost occupation tightly correlated with the flowering agave peak bloom for the region, with the population quickly dropping off at the same time that the agave bloom ended annually. See that? And we look at this table, we can see a similar shift annually of occupation with the population peak coinciding with the peak of agave bloom. So overall, roost population phenology seems to shift with agave bloom. Nectar availability plays an important role in this shift, and overall evidence for long-nosed bat tracking agave nectar resource availability in the northern distribution of the range does exist. It seems that the bats do leave when the agave are done, and they may show up early, but then head out if there's no agave blooming and come back. They seem to be very tightly tracking this resource. So although low in numbers, bats are still detected in this region in early June and late October when there are no known agave are in bloom. So this suggests that fewer nectar resources are available to the bats during these times and indicates a change in resource use between seasons. Although long-nosed bats are traditionally identified as nectarivores, former studies have indicated that long-nosed bats consume arthropods in low amounts, often reporting intake as incidental consumption while feeding on nectar or while grooming. The highest documented rate of consumption from previous work is 28% here, done by Sanchez and Median, which occurred in central Mexico. However, the majority of these studies took place in the southern distribution of their range, and our work has indicated that insect consumption may be higher in their northern distribution. And with the lack of nectar resources available during periods of occupation, it's possible long-nosed bats may exhibit diaplasticity at the northern extent of their range. So to evaluate the long-nosed bat's potential for diaplasticity, we asked how does the diet of lesser long-nosed bats vary seasonally and annually in relation to nectar availability and regional drought conditions. So we suspect that the diet between times of high and low agave nectar availability such that bats may be supplementing their diet with arthropods in periods of low flowering agave availability. So as agave nectar availability decreases, then arthropod consumption would increase. Yeah. So fecal samples were collected from bats in hand that were captured using mist nets deployed just outside the roost. So we'd set up a net right here, catch bats in hands and collect samples from known individuals. And we collected fecal samples weekly from a plastic ground sheet deployed near the entrance of the big hatchet roost and from capture. Those are captured individuals, and this is from the samples that were collected near the entrance. A subset of these samples were sent to Pisces Molecular Lab in Boulder, Colorado for genetic analysis to both confirm the bat host species and identify the plant and arthropod species in their diet. Our samples were analyzed using a technique called DNA barcoding. DNA barcoding is a method of species identification using a short section of DNA from a specific gene or genes. DNA is extracted from fecal samples and then amplified using polymerase chain reactions known as PCR. The extracted DNA is then amplified using specific primers to mark regions of DNA barcode. The marker regions are sequenced and then processed using bioinformatic analysis. In the end, we received a spreadsheet of identified species or higher taxonomic levels and numbers of reads per sample. So what are read counts? They're the number of matching sequences recorded during sequencing. They provide some insight into the abundance, but can also be generated due to variation in PCR efficiency. So typically read count data is converted to presence absence data. However, it still does provide us with some information, so we will be exploring it a little bit in this talk. So for the 4 years of the study from 2020 to 2023, we collected, turned in, and were returned 184 uncontaminated samples. So some of our samples were contaminated and had to be discarded, but our number was 184. And we looked for bat host when it was from the sheet, unless it was a bat in hand. We didn't confirm that because we had it in hand. For plant species and arthropod species. So throughout that time, from 2020 to 2023, 180 of those 184 fecal samples tested contained DNA reads from the Asparagaceae family. And we deduced, because of morphology or how nectar is accessed from those plants or when it's produced, that those plants were the agave species. And those are shown here in green. That's 97.8% of our samples tested had agave present. So plant species other than the agave are shown in gray, and they can be explained via their dominant pollen dispersal method. So these plants disperse their pollen through wind dispersal or dispersal by birds, bees, or other arthropods, and therefore can be explained through cross-contamination from other pollinators or from incidental collection of the bat on the bat's fur while roosting or flying through wind-dispersed pollens that can be ingested by the bats while grooming. Also, I'd like to state that this is from our long-nosed bat fecal samples, but we also looked at Nectar mexicana, so the Mexican long-tongued bat, and it did have other cacti and plant species that were detected in its diet that were not detected in the long-nosed bat diet. So it seems that though there may be other options available, long-nosed bats are choosing the agave. I would also like to point out that from 2020 through 2023, 184 samples contained arthropods, which make up over 80% of our samples. Which is, when we look at previous studies, 80% may appear high. But all these previous studies also use microscopic methods to determine arthropod consumption, whereas we use DNA metabarcoding. So we didn't have to have like an obvious insect, although majority of the samples that contained insects were black. So there's where they're typically yellow splats when it contains pollen. So there were visual cues that allowed us to know that there are potentially insects in those samples, but it's possible that we had a higher detection rate because of our detection method using DNA. However, a study done by Scarlett Sellers, a previous master student in our lab group, used both DNA metabarcoding and microscopic methods. And just looking at her microscopic methods, she found 49% of the samples contain arthropods, which does indicate that a higher consumption of arthropods is occurring in New Mexico, the northern distribution of the range. To further explore this high incidence of arthropod consumption in Mexico, we looked at the breakdown of arthropods detected in long-nosed bat diet by family and found that 44% of the reeds were Lepidoptera, which primarily consists of species that would be associated with flower and could have been consumed incidentally while feeding on nectar. So we then looked a bit closer at the other families in the other 66% of the reeds and determined their potential for incidental consumption while feeding on nectar or while grooming. So to do this, we organized the arthropods into 5 categories. So the first one is flowers, the arthropods that can be found on flowers or plants and may be incidentally consumed, which included the Lepidoptera family. Our second group is the cave or bats in gray. So those are arthropods that are associated with caves or bat ectoparasites and could be consumed while grooming, so also potential incidentals. So green and gray are potential incidentals. Blue represents aquatic arthropods that spend all stages of life in water. So they would not be associated with flowers or with caves or bats. And then the other category is yellow, which includes ground-dwelling and net-winged arthropods. And then the light gray is our unknown, which are reeds that were identified to too high of a taxonomic level to be categorized. So looking at our aquatic arthropods, we found Aquaculoptera, shore flies, fish flies, freshwater pelagic copiopods, freshwater ostracods, and water mites. The aquatic arthropods were only detected in the diet during periods of drought. And we also obtained trail camera footage from the BLM that shows large numbers of long-nosed bats visiting a wildlife water trough. It's possible that while drinking, bats could consume aquatic invertebrates, either intentionally or incidentally. Either way, it seems that they are using developed water sources to supplement their diet during periods of drought. And I'd like to highlight this thing because I kind of said quick, but the aquatic arthropods that we detected we only had those detected in the diet during periods of prolonged drought. So something is happening there. For our other arthropod category, we found hide beetles, green lacewings, brown lacewings, net-winged insects, grasshoppers, common desert centipedes, and spiders. These arthropods are either ground-dwelling or flying arthropods that are not associated with flowers and therefore have a low chance of being consumed incidentally by long-nosed bats while foraging on nectar. Detection of aquatic ground-dwelling and net-winged arthropods indicates intentional consumption, since it wouldn't potentially be incidental. So in summary, in southwestern New Mexico, agave is the primary nectar plant food source found in the long-nosed bat diet. Arthropods make up a significant part of the diet of long-nosed bats in this region. Consumption of arthropods occurs at a high rate throughout occupancy, so 88.4%. Detection of aquatic, ground-dwelling, and net-winged arthropods indicates intentional consumption. An observation of long-nosed bats visiting water sources and the presence of aquatic arthropods in the diet also indicate the use of water sources during low nectar availability in drought years. We also, I didn't report it here, but we found some density dependence where there's, as the population increased, there's more, we suspect there's more competition over nectar resources. And we found that instances of arthropods in the diet increase as well. So it does seem that lesser long-nosed bats exhibit diaplasticity at the northern extent of their range, but more work has to be done to better understand what this relationship means. So our last thing we'll be going over today is disease risk. And it's what we're currently working on. So what is white nose syndrome? White nose syndrome is a fungal infection that affects hibernating bats. Pseudomonas destructans, or PD, is short for the fungus that causes white nose syndrome. The fungus attacks the bare skin of the bats while hibernating. Infected bats become more active and burn up crucial fat stores necessary to survive the winter, which ultimately ends to population declines. So white nose syndrome is a new emerging threat to Arizona bats. It's a fungal infection, and New Mexico bats as well. It's a fungal infection caused by PD, in short. When this fungus attacks the bare skin of the bats while they're hibernating, it ultimately disrupts their torpor, leading them to burn crucial fat stores necessary to survive the winter. The white nose response team has confirmed 12 species in North America that have been documented with white nose syndrome, and 7 of those species are known to be in Arizona and New Mexico. There are 10 species that have been detected with only the fungus, but are not known to get the disease, and 8 of those are also found along the Arizona-New Mexico border. So bats in Arizona and New Mexico are at risk, and there's an urgent need for proactive surveillance. This map shows the occurrence of white nose syndrome and PD in North America since its initial detection in 2006. And it's reported by county, by state, or provincial or federal, or other relevant management authorities to reflect documented presence of the fungus or the disease. And it's actually since been detected in Oregon and Nevada. Not to get too much into this, but the disease has been slowly spreading from 2006 over to current day out west, and just reached New Mexico in 2025, or New Mexico in 2024, and Arizona in 2025, 2022. And it's in the region, the Sky Island region, where these bats are present. So if we zoom in there, you can see here in the Huachuca Mountains was the detection 2025, where there is known leptonicterus roosts in this area. And our biggest concern and here in Nevada, so we have concerns about potential for the long-nosed bats, a long-distance migratory bat, to be able to spread this pathogen down into the global south. Now, although they wouldn't be susceptible, they could still be vectors. And until recently, there weren't other philostoma bats that were testing positive for PD presence, but Nevada caught a California Macrotus californis, so California leaf-nosed bat, that was positive for PD. These bats also aren't hibernating bats, so it was interesting that found that, and they're philostoma, or closely related to long-nosed bats. There's a known long-nosed bat roost located in Big Bend, Texas, that has been tested positive for PD. And there's a potential that these two roost sites, Big Hatchet and that site, are connected. So there's possibility that PD could be on a bat that is co-roosting. It could establish in the cave, and it could be onto long-nosed bat and transport it throughout its range. So how are we studying this? The first thing we've been looking at for the past several years is movement study. So at the entrance of the roost, we have an antenna system set up. So right down here, you'll see on the next slide, there's an antenna, loop antenna set up for tracking pit tag bats. So pit tags they allow us to track movement of individual long-nosed bats to active monitoring sites. We currently have that antenna system set up around the entrance with a reader, which provides us presence data of tagged bats. And we're currently working with 12 other researchers throughout the U.S. and Mexico to track bats at these sites, to maintain these sites and track them. Since from 2019 to 2025, we've tagged 283. Just this project alone has tagged 283 total individuals. We're at a transition roof, so we don't capture as many individuals as others, but we do get redetections. We've also developed a homemade pit tag reader that can be deployed on a hummingbird feeder. These bats frequent hummingbird feeders at residential areas. When we see that decline of bats at Big Hatchet, when the agave bloom declines or ends and the bats drop off, we actually see a spike of activity at residential sites at their hummingbird feeders where the bats are draining them. And they tend to stay there until mid-November in New Mexico and into December, January in places like Tucson, Arizona, feeding from these feeders. So the establishment of the collaborative pit tag detection network was made possible by efforts by Teresa Laverty to track individual leptonictus rubber movements across the United States and Mexico. Individuals were documented in New Mexico across the Animas Palencios and Big Hatchet, Little Hatchet Mountains, and Silver City area. We also found individuals associated with Big Hatchet roosts that were from other tagged at other sites, even as far as Baja. Here's a close-in zoom in of our sites that the highlight ones are ones that we monitor specifically. We collaborate with Oregon Pipe, and we're going to deploy new sites in Tucson. Here's a map that shows the connection, the movements. We have movements over 1,800 kilometers between individuals. We've also since been able to link our Big Hatchet roost with our northern sites in Silver City. As you can see, there's a lot of movement between sites, and if one site does have an establishment of PD at it, it could easily spread down into the global south. So we are currently working to monitor this activity to see the spread using non-invasive monitoring techniques as well as some bat capture techniques for PD surveillance. We're working with using NA bat high priority grid cells. We hope to go to over 20 per season. So if you don't know what NA bat is, it's a multinational, multi-agency coordinated continental bat program that has standardized data collection protocols to allow organizations to better assess population status and trends to inform responses to stressors and sustain viable bat populations. So we aim to work within this framework to contribute data as early stage fungal invasion occurs. This will include uploading capture logs from our sites, any internal roost counts that we were able to collect while conducting habitat surveys and acoustic survey data, figure out where are long-nosed bats overlapping with potential susceptible species, and what potential habitats do we need to help manage or preserve or mark as sanctuary habitats. Our current project is Understanding Bat Population Dynamics and Disease Risks, which is a conservation initiative in southern Arizona and southwestern Mexico. We have 7 major goals. We want to continue long-term monitoring of big hatchet roost. We hope to track long-nosed bat movements, document bat species and seasonality, identify critical habitats for these PD susceptible bats that need to have management protections, conduct PD surveillance, conduct roost site assessments, and we also want to build local capacity with the Tahoe Oto Nation. We're working closely with their vegetation monitoring program to do that. Here's a map of our study area with all the grit cells that we have selected throughout the region that we are working on monitoring this upcoming field season. I'd like to thank all of our field crews throughout the years that have done work on these projects and helped us collect all this data. It's been a lot of work and it's really hot and tiresome to do it, so we really appreciate it. And to thank everyone else that's assisted with this project. It's been a big effort across the board. Oh, and I can take any questions.
Okay, I'm not seeing any questions. And so we'll wrap up. I want to thank you again, Mallory, for representing the Desert Southwest CESU in our seminar series. And also want to thank Catherine Stoner from the Desert Southwest and Diana Elder from the Colorado Plateau CESU for being part of our spring webinar series this year. We really appreciate it. Again, I believe Teams will automatically send out a recording of the presentation, but today's presentation will also be posted on our CESU webinar page. I put the link in the chat, and I'll have that up by tomorrow. And I want to thank all of you for participating in today's webinar. And enjoy the rest of the week and your weekend. We'll see you sometime down the road.
Thank you.